Okay, today we are going to talk about HIV. That is a very dangerous virus, HIV, human immunodeficiency virus, which is the causative agent for the AIDS, right? A disease in which there is profound immunosuppression and a lot of, you can say, opportunistic infections appear and secondary neoplasms appear and neurological dysfunctions appear, right? So HIV is basically a very dangerous virus, right? And it destroys basically our immune system over a long period of time, and destruction of immune system or impairment, severe impairment in the immune system clinically translates into appearance of opportunistic infections and secondary neoplasias and some neurological dysfunction. Today we will be specifically focused on this virus. Later on we will study about its pathogenesis of AIDS and the clinical manifestation. Now HIV belongs to the group of viruses which is called RNA retroviruses. RNA retroviruses. Retrovirus. Right? Now before I really move forward, I would love to explain what is meant by retrovirus, right? Let's go to the basic concept. In biological world, the basic concept is that all the genetic information is usually in DNA. I'm not talking about this virus, I'm talking about generally. Generally speaking, in biological world, most of the time we find that genetic information is present in DNA, right? And then DNA is transcribed into, yes, RNA, and then RNA is translated into proteins, right? Proteins which are structural proteins and yes, functional proteins, right? So in this way structure is made and functional proteins act as enzymes and regulatory proteins, and biological world is running. This is central dogma. This is the basic principle in the biological world, right? The DNA transcribes into RNA and RNA eventually translates into proteins, and proteins are basically building blocks as well as they are the functional workhorses. But this virus is different. The genetic information about the HIV virus that does not reside into DNA, when virus enters in a cell, HIV virus when enter into cell, it brings its genetic information in the form of RNA, right? So in this virus what really happens that this virus has information as RNA, and then RNA of this virus, once this enter into our cells, this RNA is reverse transcribed into DNA. It is reverse transcribed into DNA, right? This virus has as a genetic material RNA, and their RNA, it is the reverse direction. Now standard direction is DNA to RNA, but in this particular virus, the direction is reversed. From RNA we make the DNA, and this process is called, yes, reverse transcription. And then DNA is again made into, yes, two types of RNAs, right? It can be transcribed into two types of RNA, that is, yes, genomic RNA and messenger RNA, and from messenger RNA viral proteins are made, and these proteins with the genomic RNA make the new virus, right? So due to this feature that this virus does not have DNA as its genetic material, it has genetic information in RNA, and because it is reverse transcribed, right? Reverse is also called retro, right? Retro means in back direction, right? Back gear, right? So reverse transcribed, because this virus undergoes reverse transcription process, we call it retrovirus. What we call it? Retrovirus. So why HIV virus is retrovirus? Because genetic material is present as RNA, and once this genetic information of the virus enter in our cell, it is reverse transcribed into DNA, and then DNA again is in normal fashion transcribed into RNA, and genomic RNA and messenger RNA. Now This is an unusual function, right? That you convert RNA first into DNA. And for this reverse transcribing function, we need a unique enzyme, and that enzyme is called reverse transcriptase. Very good. The enzyme which is present over here, right, which convert RNA into DNA, that is called reverse transcriptase. Reverse transcriptase. And this virus is very, very intelligent. You know how we know this is intelligent? Yes. Because it has expression like you? No. Because it has RNA and it knows that if it has to go under reverse transcription, it is an unusual step, and if it has to go under unusual step, it has to carry the enzyme itself. So this virus keep on carrying what? As I keep on carrying the marker, it is my business, right? This virus business is to start the action with reverse transcription, so it keep on carrying which enzyme? Reverse transcriptase. So it's one of the very big hallmark of this RNA virus, that this reverse transcribing virus has its personal and unique what? Enzyme. What is this enzyme? Reverse transcriptase, right? This was a little introduction. Now, one question. How many people approximately are now living with HIV virus or AIDS in the world on global level? We are just going to talk about this virus, a little idea that how many people? 35 million. I think you are sitting and counting all the time. He is right. About approximately 33, 35 million people presently in the world are living with HIV and AIDS. About 33 to 35 millions, right? You know what is million, right? So 33 million people are living presently with HIV and AIDS. And approximately, how many people are dying with this disease? Average year, approximately 2 million. 2 million people die almost. I mean, no one is sitting and counting it every case, right? But approximately 2 million people die average year due to AIDS, right? Which is of course due to this virus. And so you can say 2 million people unfortunately die, right? Average year approximately, right? And average year. And how many new infections occur approximately average year? How many unfortunate people who get this virus? Yes. Anyone has some idea? It's a very sad situation that 33 million people are already living with this virus, right? And many of them have already developed AIDS also. So there are many people who are living with this virus, and almost 2 million people every year die of this complications related with this virus or disease related with this virus. And every year we get about 2.5 million new cases. 2.5 million new cases every year. So actually, while we are having this lecture, we are in the center of this tragic pandemic, most feared pandemic in the world, right? This is still spreading, right? And every year about 2.5 million people get the new infection of HIV. And out of these all 33 million people, most of the people are where? Africa. About 70% of these approximately are in Africa, right? And 20% are in Asia, and 10% is rest of the world. So you can say about 90% of these 33 million people who are living with the HIV virus or AIDS, right? Unfortunately, they are residing in developing country, or they are living in that part of the world which is having limited resources to manage such a dangerous situation, right? Anyway, so we'll go in detail later. Let's start talking about the structure of this virus, right? That how is the structure of this virus? This virus basically lives in a bubble, right? This virus lives in a bubble. That bubble is made of lipid bilayer, right? Any virus which has a lipid bilayer cover, or any virus which is having lipid bilayer cover over it, we call such virus enveloped virus, right? So basically, I will start from outside to inside, right? So if you look at the virus, if you start studying the virus from outside, right? So what is there? That there is a, yes, lipid bilayer. This is lipid bilayer, and this virus, yes, this virus is inside. Actually, the main structure of this virus is inside this lipid bilayer bubble, right? It lives inside the bubble. From where the virus gets this lipid bilayer? This is stolen material. As you go to someone's home and steal their clothes and come out, right? Yes, that is true. It's a sort of thief. That let's suppose this is a cell, and if virus enter here, right? When virus will come out, suppose this is the major viral structure. When virus will come out, right? What it will do? It will steal the part of a membrane of the host cell, right? When this virus enter into a cell and replicates, and when replicated virus is budding out, right? It is stealing the covering from the host cell, right? So even some virus don't have any moral values, right? So it takes away the lipid membrane from the host cell. And in the lipid membrane, it is having very special proteins, right? Those proteins are something like this. Yes. This is a protein, right? This is transmembrane protein because it passes through the membrane, and at the top of that transmembrane protein, there is, yes, another protein. Yes. Now, in the membrane, these are special proteins, right? This is transmembrane protein, and this is the docking protein because it adjusts with the target cell receptor. Now, these two proteins are very important, and they are having different functions. And where this protein is called, okay, this protein, I'll make it a simple structure so you don't get confused. Okay, this protein, it is called 41GP. GP stands for what? General physicians? Glycoproteins. This transmembrane protein is 41 glycoprotein, and that one is called, yes, 120 glycoprotein. And together they are called 160. That is not simple addition of course, apparently you would say 120 plus 41 is equal to 161, but because virus has not studied the mathematics, so this protein complex as a total is called 160 glycoprotein. Later on we'll discuss this, but at this very moment I want to say that viruses, major structure of the virus is inside this bubble. Bubble is made of lipid bilayer, and on the lipid bilayer, what is this being expressed? GP 41, yes, keep on saying as I'm drawing. GP, say loudly, I don't hear. GP 41, GP 41. What is the importance of GP 41? Yes, please. What is the importance of GP 41? Anyone in this room? Okay, this is part of the structure. My question is that, what is the importance of GP 41? Or just Dr. Najeeb got mad and he is trying to put the stress on some very small thing? No, it has a very important clinical importance. GP 41. Anyone? Yeah? There is some clinical importance about GP 41. Very good. There are some special drugs against it. Right? That is why I want to drill in your mind and put GP 41. Because GP 41 is a protein which help in ultimate fusion of virus lipid membrane with the our cell lipid membrane. And that is a deadly fusion. When virus membrane fuses with our T cell membrane, or virus membrane fuses with our what? Macro Macrophage, T cell, T. Right? T for?
Thymus.
Thymus, yes. Don't think wrong things. Okay. So what I'm saying, that basically this protein is not for attachment. First, virus should get attached with our target cell, right? Attachment function is done by this naughty man, and after that this protein come into action, right? And fuses the membrane. I will talk about that in a few minutes. So this is all GP, yes, 41, right? And what is this? GP? 120. GP 120. What is the function of GP 120? Yes? It helps the virus to attach with the target receptors. What is this again? GP 120. Now, I will tell you exactly how these two proteins are interacting and helping the virus to have a deadly handshake with our cell. How? Actually, I think virus heard it. Right, so we were talking about that what is the role of GP120 and GP41, right? Let me tell you. This virus loves to infect CD4 positive cells. This virus loves to infect CD4 positive cell, right? Now CD4 positive cell, let's talk what are the cells which are CD4 positive, right? Because this GP120 loves to bind with CD4 molecules, right? Let's suppose this is here our one cell, and if this cell has a molecule here, and this molecule is CD4 positive, right? If this cell is having this receptor, then this virus can attach with that cell, right? And how this virus can attach with that cell? By the GP120, right? Binding at which point? At CD4. Is that right? It means that primary, listen now carefully, the primary attachment between the virus and the target cell is the interaction between GP120 and CD4. So we must know which are the cells which are CD4 positive, because those cells will be the target of this virus. Is that right? Am I right? So I will tell you one cell which is CD4 positive, that is T helper cell. One cell I am telling you, T helper cell, they are CD4 positive. Other CD4 positive cells, you will tell me, yes. Number one, T helper cell is CD4 positive. Then other CD4 positive cell is, very good, monocytes. Monocytes, these are circulating in the blood. Then there are, when monocytes enter into tissue and monocytes become activated, we call them macrophages. Monocytes are also having CD4 molecules. Macrophage also have CD4 molecules. Even some of the macrophages, they enter into central nervous system, and those macrophages are called microglia. In the central nervous system, there are some monocyte derived cells which are called microglia. They are also having which molecules on them? CD4. So we are talking about CD4 positive cells. So classically an average student will tell that T helper cells are CD4 positive. Of course that is true. But a very good student knows that there are other cells which are also CD4 positive. What are the other cells which are CD4 positive? Monocytes, macrophages, microglia, and then there are special dendritic cell under the skin. Under the skin, there are special dendritic cells, right? They are also having CD4 molecules. These dendritic cells which are under the skin, they are having a special unique name. Yes? Langerhans. Very good. And Then there are other dendritic cells which are present in follicles residing in the spleen and lymph nodes. They are called follicular dendritic cells. They are called follicular dendritic cells. They are in spleen or they are in follicular dendritic cell or in the lymph nodes. Now, why I mentioned these cells? Now, how many cells we have which are CD4 positive? One is T helper cell, number 2 is monocyte, third is macros, fourth is micro, glia, fifth is Langerhans, and sixth is follicular dendritic cells. So HIV virus loves to bind, rather loves to attach with these 6 type of cells. Is that right? This virus loves to attach with these 6 cells specially. Why? Because these cells are having CD4 positive molecule. And what really happens, there is a handshake between GP120 and it loves to bind with what? CD4 molecule. Is that right? Now another thing. Let's suppose that, okay, I put one CD4 positive cell here and we see how the attachment proceeds. If there is CD4 positive cell here, let's suppose this is CD4 positive cell, it may be T helper cell or macro. Now, here is our virus. Here is our virus, and virus is having what protein is this? Yes, GP41. Say loudly. And what is this? GP120. And GP120 is going to bind with which receptor? CD4. So they will engage. This is called primary engagement. What is it called? Primary engagement, right? And this process is called primary attachment. But it is not enough. You know, this virus is something like boys. You know boys, if girl give her a hand, what is the next step? They will try to bring other hand also somewhere. Same is true about this virus. Yeah, that is true, everyone knows, right? This class is only for adults. Now, what happened? As soon as, actually previously it was very sad. But now it is happy because it is engaging with CD4 molecule. The moment it engages with CD4, let's suppose now it is engaged, and what molecule is here? CD4, and this is the cell. Is that right? Immediately it will bring, previously it was having 2 hands. Okay, you imagine. And these hands are in the pockets, not active. Right when it was sad. You know, like that. The moment there is action, one hand will come out. Right? Now what this hand is doing, it is also a very important thing. Hands are important. This hand will come out. When it will come out, it will be expressing itself like that. And other hand will come out too, like that. Now, what is the purpose of these hands? Actually, this molecule, GP41 molecule, okay, very good, it means you are listening, great. GP120 molecule, very good. Once it interacts with CD4, it undergoes a conformational change and it expresses 2 more, they don't call hands, but anyway, they expresses 2 more active sites, and now they would love to interact with some other place with that target cell. Is that right? Now what is the other place? Target cells are also clever, you know, they provide other place. Actually, target cell, depending upon which target cell. If it is let's suppose if it is macrophage, suppose it is macrophage, right? Then, as soon as this hand come out, this will interact with another protein, and this protein is called, this is actually a physiological receptor for chemokines. Of course, our cells are not designed to welcome the virus. These receptors were actually designed for some other immunological function, but unfortunately virus is taking advantage of the situation. So what really happens, that first GP120 binds with CD4, and after that GP120 undergoes conformational change, and then it expresses another arm which will interact with a co-receptor. What is this? Co-receptor. This co-receptor is actually a receptor for chemokines. It is a receptor. Chemokine is also a type of cytokine, of course. Chemokine, right? Now, if it is macrophage, right, then this receptor is called, yes, this chemokine receptor, this is called CCR5. Chemokine receptor number 5. So what happens? That your antigen presenting cells, especially macrophage, monocytes, microglia, dendritic cells, they are not only having CD4, but they are also having CCR5, and virus, once it is activated GP120, right, it expresses a domain which interact with the CCR5. Is that right? Once, or other chances that rather than macrophage, if it is T helper cell, then mechanism is a little different. How? That of course T helper cell also has CD4, but it does not have CCR5. T helper cell does not have CCR5. It has other type of chemokine receptor, and this chemokine receptor is CXCR4. What is it? CXCR4. Now, what is this? This is on the T helper cell. Now, okay, I will make it on the other side. You will know why I have made it on the other side. Because when virus comes here, let's suppose I'm repeating this virus structure here. When virus is coming here, here it was, yes, what was this protein, please? GP41, and what is this? GP? Happy. It brings one arm out. This arm is CCR5. Can it engage here? No, because T helper cell does not have CCR5. But then it will bring other hand, and other hand is capable of interacting with CR4. Right? Am I clear? Now, so this is the importance of GP120. That number one, it is the primary docking protein to dock or attach with the target cell. And which target cells? Which are CD4 positive. But for real attachment, it also requires co-receptor interaction. Co-receptor is what? Chemokine receptors. Chemokine receptors which are present on macrophages are CCR5. Co-receptors which are present on T helper cell is CXCR4. Once primary attachment is done and also co-receptor is activated, then is the real bliss, happiness. What? That now when 120 undergo change and it brings a special signal to, what is this red molecule? GP41. Once both of them are attached, then GP41 will slip on the side. It will slip on the side, undergo a change, and hook the membrane of the target cell. You understand what's going to happen now? That GP120 had only primary and secondary attachment. The real action is started by which? GP41. Once these two attachments are done, as I told you, boy and the girl, primary first and second attachment has to be there. If there's no attachment, no action. But now the real action comes. What is this? GP41. And GP41 will become super active, slip on the side, and then hangs the membrane. Enters into membrane. And then start pulling this membrane towards virus membrane and start pulling the viral membrane towards the target cell.